Look at the energy signature around the mitochondria. The folding is unprecedented.
It's stabilizing. We should initiate the sequence protocol immediately.
Look at the replication rate in dish 4. It's exceeding projections.
Adjusting the nutrient flow now to compensate.
See how the GFP tag illuminates the entire structure. That confirms the localization we were looking for.
The structural anomaly is stabilizing.
This sequence here, it shouldn't exist. This changes everything, doesn't it?
Purity levels holding steady at 99.997%.
Tablet confirms. Scrubber array appears stable.
Levels are rising in Sector 4.
Reroute the samplers. Analyze the spike immediately.
Seal secure on the hood.
Check.
Gloves double sealed. Ready for entry.
The sample transfer is complete for stage 1. The viability reading is stable.
Acknowledged. We can proceed with the incubation sequence.
Look at the morphology here at the apex.
It appears stable. The structural integrity is holding up under rotation.
This confirms it. The regulatory elements are fully expressed.
We are seeing the complete genetic blueprint unfold in real time.
People. Using this advanced micromanipulation system, we can isolate the healthiest cells with unparalleled precision.
The selection process requires absolute precision. Only the healthiest cells, identified by the glowing...
The markers are stable across all optimal candidates.
Confirmed. Proceed with isolation sequence 7.
Cross-reference the humidity reading with the tablet data.
Looks consistent. The environment is stable.
The furrow is beginning to form exactly as predicted.
Noted. Initiating high-resolution scan sequence 7.
The stabilization sequence is nearly complete.
Run the high-resolution capture protocol now.
The replication rate is accelerating exactly as hypothesized.
The structural integrity remains stable at 4K resolution.
Look at the oxygen levels stabilize here.
Yes, the adjustment to the gas input was successful.
Look at the proliferation rate on dish 7B. It's exceeding the projected metrics.
Confirmed. Adjusting the parameters now. The division is accelerating.
The proliferation rate in this collis rate moving.
The
AI is showing a significant spike in the division rate here.
Yes, the cellular acceleration is unprecedented in this sample.
The symmetrical growth pattern is holding steady.
Yes, the data confirms healthy development across all samples.
Look at this cluster on the right. The morphology is completely irregular.
Agreed. Isolate sample 4B immediately.
The aspiration pressure is holding steady.
Good. We have clean separation. Move to containment.
Confirming passage complete on tray 4.
Plating density looks optimal.
Look at the expansion in tray 4. The colonies are stabilizing faster than expected.
Agreed. The new protocol is definitely working.
The migration pattern is accelerating. Yes, the signaling molecules are having the desired effect.
Look at the morphology on screen 3. The elongation is stabilizing.
Yes, the actin filaments are reacting strongly to the new medium.
Look at the spike in the BCL2L1 gene expression.
Confirming new activation sequence detected at 4Q21. It matches the profile.
The cellular response in quadrant 3 is significantly higher than the baseline.
Yes, the signal appears to be working exactly as hypothesized.
Look at the diffusion in the lower quadrant.
Yes, the saturation is consistent with the projected model.
The markers are attaching exactly as expected.
Look at the cluster forming on the P53 pathways.
Focus right here on the red cluster. The movement seems faster.
Adjusting the Z-stack now to compensate.
Look at the red cluster in compartment 4.
The signal intensity is off the charts. Cross-reference with the gene expression data.
See how the markers are moving along the neural pathways.
Yes, the speed suggests a reaction to the compound we introduced earlier.
Look at the interaction points here.
The binding affinity is stronger than we projected in the simulation.
The fluorescent markers are stable across the time lapse.
Yes, the mitosis sequence is confirmed and archived.
The aggregation pattern here is unexpected at this concentration.
Let's cross-reference the cellular protein activity logs from the automated system.
There, position 427. Do you see the translocation?
I'm isolating the segment now. It looks novel.
The analysis confirms the deletion on exon 12. That explains the phenotypic data we observed.
Yes, the sequence alignment clearly shows the frameshift mutation.
Look at the highlighted section here. The robotic analysis confirms the variance.
I see. The control group shows no such anomaly.
The mitochondria are reacting strongly to the adjustment, and the division rate is accelerating. The data confirms the hypothesis.
The diffusion rate appears optimal at this concentration.
Agreed. The signaling pathway is activating as expected.
The H12 culture is decelerating significantly at 48 hours.
While the control group expands exponentially. Adjusting cytokine levels now.
Look at the trajectory of this specific clone.
The growth rate is significantly higher under condition 7.
Yes, the 4K imaging confirms the increased viability.
These patterns correlate exactly with the sequencing data.
And the cellular development confirms the hypothesis.
The biological data stream looks stable now.
Yes, the automated analysis is confirming the success rate we expected.
Look at the correlation here. The AI found a pathway we never anticipated.
It's completely rewriting the known cellular response model. This changes everything.
Cross-reference the analysis with the updated protocols. We need to ensure the parameters are exact.
Ensure the robotic arm calibrates before the next sequence.
It's already done. The data is updating on screen 3 now.
Temperature and gas balance are stable across all units.
Confirmed. The robotic inception was successful. The growth pattern here is unusual for this lineage. I agree. The software flagged it an hour ago. Let's isolate the data.
The sequencing data on run 7 looks anomalous.
Cross-reference it with the microscope feed from Incubator 3.
All parameters are within tolerance according to the environmental logs.
Yes, the verification is complete. I'll sign off now.
The cell structure looks stable at 400x magnification.
Cross-reference that with the gene sequence analysis.
In the realm of advanced biotechnology, precision is paramount. Here, automated systems.
The accumulation data confirms the structural change in the 5G helix.
Cross-reference with the sequencing report from week 3.
The automated system highlighted this anomaly in the archive data. Yes, the variance is statistically significant compared to the current findings.
Look at the correlation between the gene expression and the cellular morphology here.
Yes, the p-values are significant across the board.
This segment here, the alignment is undeniable.
The molecular processes are matching the models perfectly.
Look at the spike in the red channel here. That shouldn't be possible.
Yeah. Zoom in closer on sector 4. The morphology is completely irregular. Look at how the mTOR pathway is reacting to the compound.
It's stabilizing the neural network.
This changes everything.
Based on the new data, this pathway is the most promising.
Confirming the sequence now. The automated calibration is complete.
The sequencing results came back positive on this run.
Good. Let's monitor the incubator levels and prep the next samples.
This graph indicates a thermal imbalance in the main chamber.
I see it. Increasing the coolant flow rate by 5%.
The Q3 timeline projection looks promising.
Yes, the cellular degradation rate has stabilized exactly as predicted.
Look at this data point here. The improvement is marginal but significant.
I agree. This confirms our hypothesis moving forward.
Zoom in on the molecular diagram for segment 7.
The incremental changes are visible now. We have a breakthrough.
The resolution on this imaging component is exceptional.
Yes, the calibration sequence looks stable now.
As you can see on the main display, the cellular regeneration markers are highly active across all samples. If we rotate the visualization here, the anomaly becomes much clearer.
I see it. Let's isolate that specific cluster for further analysis.
If we adjust the molecular structure here, the reaction rate increases.
But what about the stability parameters from the Tokyo feed?
Look at the luminescence in plate 3.
The automated system is tracking the kinetic response now.
This facility represents the next stage of discovery. integrating AI and human expertise.
I'm adjusting the focus on the outer ring now.
The expansion here is significant since the last reading.
We should log the time-lapse data immediately.
The growth curve on the left dish shows a significant acceleration at the 48-hour mark.
Yes, the molecular activity confirms the spike we predicted.
Zoom in on the mitochondria activation.
The ATP production is accelerating faster than predicted.
Cross-reference that against the control data.
The ER network seems significantly more dense in this sample.
Agreed. The interaction with the mitochondria is clearly visible now.
The visualization clearly shows the molecular binding site.
Can you rotate the complex so we can see the full pathway interaction?
The flow cytometry data confirms the activation markers.
Good, let's cross-reference with the RNA sequencing results.
Look at the spikes in the Q3 data from 2042.
Yes, the environmental stabilizers show a direct response to the neural inputs here.
The simulation confirms the pathway. The reaction rate is significant.
We should cross-reference the RNA sequencing data immediately.
Zoom in on the endoplasmic reticulum, and let's analyze the protein folding.
Remarkable detail. The simulation correlates with the in vitro data.
If we adjust the parameters here, the robotic arm can synthesize the compound faster.
Yes, and the data suggests...
Character C, have you logged the new parameters for the 4B sequence?
Yes, doctor. The system is initiating the transfer now.
The mitochondria show increased activity in the green spectrum.
Yes, indicating high energy transfer. Note the structural changes here.